Okay, I'm going to give you a little guidance on how to do a good job of short answer questions on the exam. So, first, focused. Just answer the question. Avoid including irrelevant material because you'll get marked off for including things that aren't at all relevant because it means that you think they're relevant and they're not. The answers should be precise, and by that I mean you should use strong. Don't be vague when you can be specific. And I'm going to post a couple of good concept maps that demonstrate that as well. Your answers should be logical. So, for example, if it's a question that describes a process or an experiment, or a molecular pathway, if you have all the steps but they're in the wrong order, you're not evidencing strong conceptual understanding. So that's important as well. And then in all scientific writing, but particularly in exam answers, aim for concise. Don't aim for fancy. Avoid long sentences with lots of commas in them. Short, clear, to the point is what we're aiming for. And some questions will actually tell you how many sentences are in the key. And then finally, if we ask for sentences, which I often will, don't provide bullet points because bullet points don't give me a chance to really assess your understanding of certain concepts because they're so short. Sometimes I will ask for bullet points. So make sure you're giving me what I ask for. Okay, so to do this, which I've never done before, I'm going to use an old exam 1 question about marker rescue because you've had that material. And so here's the question. Some class discussion has focused on how an experimentalist would know that a phenotype is due to a specific mutation. And one approach for either genetically engineered mutations or classically selected mutations is to repeat the construction or selection and see if the phenotype is stable. Another approach is referred to as marker rescue. So this question has 2 parts, and I'll read you the 2 parts, and then we'll go through some sample answers, some real answers. So part A, for 2 points, for a DNA virus like HSV1, explain how you would use marker rescue with a wild type HSV GC glycoprotein C gene DNA fragment that has flanking sequences and presumably mutant viral DNA to show that the enhanced spread in the mutant virus was actually due to the mutation within the GC gene. And part B, for 3 points, explain the important distinction between marker rescue and complementation. And remember, this also relates to a picture of GC spread mutant plaques that I showed this week in class, and then the approach that was in the genetics and genomes lecture. Okay, so we'll look at some answers, some student answers from last year that I have approval to use to improve teaching and learning for the answer to part A. So, for a DNA virus like HSV1, tell me how you would use marker rescue with a wild-type GC DNA fragment that contains flanking sequences and presumably mutant viral DNA to show that the enhanced viral spread in the mutant virus was due to a mutation within the GC gene. And so one student said, use flanking sequences to guide transformation of the GC region into the mutant DNA to observe if the wild-type phenotype is seen after incorporation of the sequence. And that answer got full credit. It's a simple sentence, but it makes me believe that this student understands that the DNA from the fragment with its flanking sequences is going to undergo homologous recombination and recombine into the viral DNA and replace the sequence that has the mutation, replace the GC gene in the mutant viral DNA, and that the phenotype would then be presumed to be wild type. So, that student could have written a few sentences more or could have embellished this, but when I read that simple sentence, I believe that student understands the relevant biology. So a second answer here that I'm sharing with you is transfecting the original wild type to the mutant, and if the mutation is not observed. So this answer is using too few words so that I'm not sure what they understand. So transfecting the original wild type what to the mutant, to the whole virus. I'm just not clear about the front part of that sentence. And if the mutation is not observed. So that suggests to me that the student is thinking that I would look at the genotype, because a mutation is on a gene, not a protein, and they didn't use the word phenotype. So this is lacking in the precision and clarity. And then the third, and that got no points. And then the third answer, and these were graded by the grader, but I don't disagree. And the third answer was, first you replace the wild type HSV-GC gene with a selectable marker like LAC-Z and select colonies that have been successfully transformed, blue in this case. Then you replace the LAC-Z gene with the mutant gene and select the white colonies and then purify the virus from those colonies and infect permissive cells and study spread in those cells. So what this student has done is repeat parts of what they understood from the protocol for making the mutant in the first place, but that wasn't in the question. I did talk about that, but it wasn't in this question. I was asking for something after that to see if a student understood marker transfer, to understand if a particular gene is responsible for a mutation. So that got no credit either. So, if you should practice writing answers to these questions. Make sure you really understand the answers, and then it doesn't have to be long. Okay, so we're going to do the second part. So this I gave you a bunch of different answers, possible answers, and this was worth 3 points. Explain the important distinction between marker rescue and complementation. So the first one says, complementation is when the gene products of two mutants work in tandem and result in the wild type phenotype. Marker rescue is a mutation on one gene and that does not possess mutant parents. So this answer is good in that it's answering both parts, telling me what the two different processes are. But the answer to the second part, marker rescue, is a mutation on one gene and does not possess mutant parents, is not correct. So this got half credit. Here's another one. Marker rescue would indicate a change in the genotype, but complementation is a phenotypic change, not due to a change in genotype. And that is true, but what this answer doesn't do is tell me that complementation gives you a wild type phenotype because proteins from the two parents are able to complement the deficiencies, the mutations in the parents. So it shows that the mutations are on two different genes. So that got half credit. And again, these were scored by the grader. Third one, complementation yields mutants similar to parents. Marker yields different progeny from parents. So again, this is sort of mushy. I think the student understands that one is a phenotypic change and one is a genotypic change, but complementation doesn't yields mutants. So the two mutants are there. It's this grader gave this a 1. I might have given it a 1.5, but it's not complete. It's not explanatory. Here's the fourth one. Complementation is when a virus cannot produce a viable protein, and so another complements it, misspelled, by producing a viable protein for both. And that is true, but where is the other half of this? So that's missing pieces. Here's the next one. In marker rescue, you are taking pieces of DNA and creating a new mutant product different from the original strands. And that's not true because marker rescue in the experiment that I described takes a wild type fragment and replaces a mutant gene. Whereas in complementation, the progeny cells, we're not talking about cells, so that's wrong, imprecise word, are either of the two parental phenotypes and not something new and different. And so that's kind of getting at it, but it's not explaining what's really happening. And then the last two got full credit. Marker rescue involves homologous recombination between linear DNA and mutant viral genes. The particles expressed are fully infectious as the genomes they hold have all the tools for infection. In contrast, complementation is when two viral genomes with separate mutations can together encode all the products necessary to produce mature virions. However, the genomes of the progeny haven't changed, so if they were isolated, each particle on its own would be incapable of producing a plaque. And that got full credit. And that one has a lot of words, but I absolutely understand that I absolutely believe that the student understands what the inherent difference is in the context of the scenario set forth in the question. And then the last one, complementation does not involve recombination because the gene products from two infectious virions come together to permit infection. In marker rescue, recombination occurs. And that's to the point. It doesn't have as many words. In this case, the student used the word infectious virions. Yes, they are able to infect the cell. They wouldn't independently form plaques because they each independently have mutations, but when they co-infect a cell, the defective gene products are complemented by the other. So, you can see the scoring is somewhat subjective. We know what we are looking for, but it doesn't have to be long. You really need to evidence what you understand. So make sure your sentences are nice and clear. So I hope that's been helpful. Thanks.